Hello everyone. Welcome you all for the lecture on anaerobic digestion for biogas production in the subject biomass and bioenergy. You all know that lots of waste are being generated from the industry as well as from the domestic. These waste can be organic or inorganic in nature. If these waste are left as such and dumped, it's going to create lot of environmental problems. So we need to have a right technology to treat this waste, and to make the technology commercially viable, along with this waste treatment, we need to extract the valuable compounds out of it. In today's lecture, we are going to see one such technology called anaerobic digestion for wastewater treatment as well as for the biogas production. Let us get into the lecture. Now you can see a picture. In this picture, you can see lot of bubbles are coming out from the sewage. You all know how these bubbles are produced. These are due to the microbial action. The microbes present in the sewage consumes all the organic waste and it produces the lot of gases. These gases you can call it as methane gas, or simply you can call it as swamp gas. This you can see it in all kinds of wastewater treatment plants. As a biotechnologist, you need to take this nature's information and you have to develop this as a technology in the lab for the biogas production. So at the end of this today's lecture, you can able to apply the process of anaerobic digestion for the conversion of waste biomass to biogas. This will be accomplished through three specific objectives. In the first specific objective, you can able to indicate the different waste biomass and the microbial consortia that is utilized for the biogas production. In the second specific objective, you can able to illustrate the operating parameters that we need to keep it for the anaerobic digestion. In the third specific objective, you can able to demonstrate the process of anaerobic digestion for the biogas production. So before getting in depth concept of anaerobic digestion and biogas production, we need to know some basics about the biogas and its importance. Biogas is a natural energy rich gas. It composed of majorly these three type of gases: methane, and then carbon dioxide, and then hydrogen sulfide. In these three gases, methane comprises the maximum of 60 to 75%, and next comes the carbon dioxide, which is 20 to 25%, and the least one is the hydrogen sulfide, which is 1 to 3%. You all know that carbon dioxide is commercially utilized as a fire extinguisher. This carbon dioxide is present in the biogas. It's going to reduce the calorific value of the biogas. So we need to remove this carbon dioxide. Similarly, whenever you are crossing the sewage water treatment plants, you can smell the bad odor. This bad odor is because of this hydrogen sulfide. Moreover, this hydrogen sulfide is corrosive in nature and it will corrode all the metal parts that we are going to have it in the reactor for the biogas production. So we need to remove this hydrogen sulfide also. So when this carbon dioxide and hydrogen sulfide is removed from the biogas, then this biogas will become high calorific value gas, and this we call it as upgraded one or the biomethane. Now we will see why we need to produce this biogas. We all know that for our day-to-day activities like for transportation, cooking and electricity generation, we are depending on the fossil fuels like natural gas, oil and then coal. These fossil fuels are non-renewable energy sources. That means if we completely use it, we cannot get it back. So we need to go and search for the alternative energy sources. There are n number of energy sources that is available on earth. Some of them are solar energy, wind energy, biomass energy, hydrogen energy, geothermal energy, ocean energy and then hydropower energy. So these energies are renewable energies. So that means we can continuously extract the energy from these kind of sources. The biogas that we are seeing in today's lecture comes under this biomass energy. You all know in the future all our cars are going to run in the electricity or through biogas. This is because renewable energies are going to be the cleaner fuel, and then it won't produce any residues, and it is a non-polluting, and it won't produce any smoke, and moreover it is economical. So we need to search for alternative energy sources. For that purpose only we are studying in today's lecture about the biogas production. Now we will see how this biogas is made. As I told you in the start of the lecture, you can use any type of organic waste for the production of biogas. Say for example, you can use sewage waste, you can use municipal waste, you can use animal residues, agricultural residues, forestry residues, and then industrial residues. And these residues you need to take and you need to analyze its composition before employing it for the biogas production. And once you collect all this waste will be taken up by the group of bacteria that is called anaerobic bacteria. And the complete environment what we need to maintain is the anaerobic environment. That means there should not be any oxygen's presence. This total process we call it as anaerobic digestion. You can see it in this short video that lot of waste are being collected and then it is given to the reactor that we call it as anaerobic digester. In that the organic waste are decomposed and it produces the biogas. So once you produce the biogas, you need to know where you are going to apply this biogas. So biogas can be applied in different kinds of fields. One is for the electricity generation, and second one is for cooking, and third one is it can be used for the transportation. So for this purpose, we need to work interdisciplinary with mechanical engineers as well as with the electrical engineers for the production of different kinds of modified engines as well as for the modified generators. Now we will see the operating parameters involved in the biogas production. First is the reactor. As you all know that the complete process is anaerobic, there is no oxygen should present. So the tank should be completely sealed and it should be free of oxygen. The reactor we call it as anaerobic digester. Next is the microbial source that we are going to utilize for the biogas production. The main microbial source utilized for biogas production is the bacteria. It is not the single bacteria involved in the anaerobic digestion. It is a group of bacteria which will carry out the anaerobic digestion and converts the organic waste into biogas. Next is the carbon to nitrogen ratio. For the better growth of the bacteria, it requires the proper amount of carbon and nitrogen. Nitrogen is the limiting factor. Once the nitrogen exhausts from the system, bacteria will not go and consume the carbon. If bacteria doesn't grow, then ultimately biogas will not get produced. So we need to maintain the proper amount of carbon to nitrogen ratio. So the optimum range is between 20:1 to 35:1. So the optimum range for this biogas production that we are maintaining is 30:1. Next is the solid to water ratio. Equal amount of solid and water should be mixed and it should be made into slurry. This is because the bacteria can easily move around and then it can take up the organic waste and it decomposes it and produces the biogas. So for that purpose, we need to equally mix the water and the solid waste. Next we see about the pH. The pH for the biogas production should be maintained at the neutral condition. This is because during the process of anaerobic digestion, lots of acids will get produced. Due to this, the system's pH will go down. So when the pH gets down, this will not allow the bacteria to grow. So to avoid that, if you maintain the pH at the neutral condition, it will enhance the bacterial growth and ultimately it results in the higher amount of biogas production. Next is the temperature. The group of bacteria involved in the anaerobic digestion is in the mesophilic or thermophilic range. That means you have to maintain the temperature of 30 to 60 degrees Celsius in the anaerobic digester. So we need to insulate it and maintain the temperature. Next is the process. If you look at the process, there are two processes. One is the batch and another one is the continuous. Batch means there will be a single input and there will be a single output. In case of continuous, you can continuously feed the organic waste and you can continuously get the biogas production. And next is the residence time. So once you put the microbial source and the organic waste inside the anaerobic digester, you cannot expect the biogas immediately. The microbes will take some time for its adaptation. That time we call it as residence time. This is usually between 15 to 40 days. So once you start the process of anaerobic digestion, you can expect the production of biogas at the maximum of 41st day. Next we see the inhibition by the chemicals. We all know that the food waste and the agriculture waste that we are consuming contains lot of chemicals, and this is due to the chemical fertilizers that we are utilizing in the agriculture field. These chemicals will inhibit the bacterial growth by blocking the metabolic pathways. So we need to bring these chemicals to the threshold limit and then only we can administer for the anaerobic digestion. Otherwise, these chemicals will inhibit the growth of the bacteria and ultimately there won't be biogas production. Now we are going to see about the methanogens. Methanogens are the major group of bacteria which converts the organic waste to methane. These methanogens are prokaryotic and this belongs to the family Archaea. These methanogens are rich in the cow's ruminant as well as in the cow dung. So we need to isolate the active strain from these kind of sources and we need to utilize it for the biogas production. So these methanogens follows a process called methanogenesis for the production of methane. The methanogenesis can be of two types. One is hydrogenotrophic methanogenesis, another one is acetoclastic methanogenesis. In hydrogenotrophic methanogenesis, hydrogen and carbon dioxide combines together and it produces the methane. In case of acetoclastic methanogenesis, acetic acid breaks down to methane and carbon dioxide. Now we are going to see about the anaerobic digestion. Anaerobic digestion is not a single step process. It is a collection of steps that is involved in the conversion of organic waste into biogas. So microbes will act on the biodegradable materials in the absence of oxygen and it converts it into biogas. So if you put 1 mole of glucose, it is converted into 3 moles of carbon dioxide and then 3 moles of methane. In this, the methane is our target product. So if you put 180 grams of glucose, you can get 48 grams of methane. So this is the theoretical yield. So whenever you are working with the biogas research, you need to check whether you are getting the similar kind of yield. Now we are going to see the metabolic pathway or the biological process involved in the anaerobic digestion. So whatever the waste that we are putting inside the anaerobic digester, all this waste is going to be the polymer. Like the food waste, if you take the chapatis and buris, sambas, everything is going to be the polymer. It's going to contain carbohydrate, proteins and fats. So bacteria will not consume the polymers. So we need to convert these polymers into monomers. So for that purpose, in anaerobic digestion, there are 4 steps is followed. One is the hydrolysis, another one is the acidogenesis, another one is the acetogenesis, and finally is the methanogenesis. So in the hydrolysis, all the complex polymer is get converted into the monomers. The carbohydrates will convert it into the sugars and fats is converted into the fatty acids and then proteins converts into the amino acids. So once you get all the monomers, these monomers will be consumed by the other group of bacteria called acidogenic bacteria and the process called acidogenesis, and because of this process, all your monomers will get converted into the volatile fatty acids like propionic acid, butyric acid and valeric acid. Along with these volatile fatty acids, hydrogen sulfide, carbon dioxide and ammonia will also produce. So this volatile fatty acid acts as a substrate for the other group of bacteria called acetogenic bacteria. These acetogenic bacteria converts volatile fatty acids into majorly of acetic acid and then hydrogen and carbon dioxide. So once from this step, the pathway will split into 2 directions. One is acetoclastic methanogenesis and another one is the hydrogenotrophic methanogenesis, as we have seen in the earlier slide. So the final product is going to be the methane. Methane is of low calorific value when you compare it with the hydrogen. So researchers are trying to inhibit these methanogens so that they can stop this metabolic pathway with the hydrogen, which is of high calorific value gas. Now we are going to see about the reactor that we are going to utilize for the anaerobic digestion. So this reactor we call it as anaerobic digester, and this should be completely sealed and there should not be any oxygen inside this reactor. It is of cylindrical in shape and the top portion looks like a dome in shape. This is because the gas that is getting collected will get accumulated inside this dome. From one side, all the organic waste will get inside the anaerobic digester. So once the anaerobic decomposition is over, then the gas will get produced and it will get accumulated at the top portion. This is the headspace. So if you look at here, you can see the 3 layers. The top layer is going to be the fluid layer and the middle layer is going to be the active layer and the bottom is going to be the solid. So the middle layer is the layer in which the microorganism effectively consumes the organic waste and it produces the gases. The decomposed solids are settles at the bottom and it is removed as a sludge which is rich in the nitrogen content. The total setup of anaerobic digester should be insulated in order to maintain the temperature range of mesophilic or thermophilic, that is 30 to 60 degrees Celsius. So there are different types of anaerobic digesters are available. Some of them are given here. One is the fixed dome model, another one is the floating dome model. Next one is the upflow anaerobic sludge blanket digestion. And next one is the continuous stirred tank reactor. And next one is the two-stage anaerobic digester, then anaerobic activated sludge process. Among these kind of reactors, fixed dome model and floating dome model are commercially implemented one. So we are going to see these two models in detail. Now we will see about this fixed dome model. As the name indicates, this fixed dome model is rigid in nature and it does not contain any moving part. It majorly comprises of three parts. First one is the slurry tank, second one is the anaerobic digester, and third one is the overflow tank. Once we prepare the slurry by mixing the organic waste and the microbial culture and by adding the water, then the slurry will fed to the anaerobic digester. Inside the anaerobic digester, microbial will consume this organic waste and it will start to produce the biogas. The biogas will get accumulated at the top portion that we call it as headspace. So once the biogas gets accumulates in this headspace, it's going to exert the pressure over the surface of the slurry. Because of this pressure, the solids present at the bottom will move towards the overflow tank. And if you want to collect this biogas, there is a valve that is present over the anaerobic digester, and we can use that and we can able to collect the biogas and apply it for different applications. This model is having several advantages like less investment, less maintenance and less manpower. But it is having a risk of explosion. The pressure accumulating in the headspace, if it crosses the threshold limit, then it's going to cause the explosion. So we need to operate this fixed dome model in the presence of trained technicians. So now we are going to see the floating dome model. There is no much difference between the fixed dome model and the floating dome model. The only difference is the moving part of the gas holder that present in the anaerobic digester. So once the slurry gets into the anaerobic digester and decomposes, biogas will start to produce. Once the biogas accumulates the headspace, then the drum will automatically goes up. And if the valve above the gas holder is open, then the drum will go down. If it goes down, it will push the slurry and the solid that present at the bottom will move towards the overflow tank. Since the gas holder moves up and down, it avoids the risk of explosion. This model has certain disadvantage. It requires a lot of investment and then a lot of manpower as well as high maintenance. Now we are going to see the overall process of biogas production. First step is we need to take the organic waste and mix it with the cow dung, which is rich in the methanogens, and then we need to add the equal amount of water to make it into slurry. Once the slurry is ready, we are going to put it in the anaerobic digester. In the anaerobic digester, anaerobic digestion will take place and the organic matter will get decomposed and the biogas will start to produce, and this biogas will get collected in the gas holder. Once the biogas gets produced, then this can be applied for different applications like electricity generation, for cooking and also for transportation. Another advantage of this biogas production is the solid waste. The solid waste coming from the bottom can be utilized as a biomanure, which is rich in the nitrogen content. So it can be utilized as a biofertilizer. So if you look at this biogas production system, nothing is going as a waste. So whatever the thing that you are putting inside the digester, everything is coming out as a product. So this one we call it as a zero waste technology. Now I would like to show you a video in which you can able to understand the overall process of the anaerobic digestion. So if you look at this video, you can see the organic waste are being collected and it is pre-processed, that is the size is getting reduced and then soaked in the water and made into slurry. Then it has feed into the anaerobic digester where the anaerobic digestion will take place and the biogas will get produced. That we call it as raw biogas. Once the raw biogas is produced, it will be in low calorific value because of the presence of carbon dioxide and then hydrogen sulfide that you know very well. So we need to remove this carbon dioxide and the hydrogen sulfide by the cleaning step. This carbon dioxide can be removed by passing the gas through the water, and this process we call it as absorption, and also we can pass it through the algal cultures which can take up this carbon dioxide. This hydrogen sulfide can be removed by using some kind of solvents as well as some catalyst. So once this carbon dioxide and then hydrogen sulfide is removed, then the biogas will turn into biomethane, which will be of high calorific value. So now we will see some of the advantage or the benefits which we can get from the biogas. First is the replacement of fossil fuels. So we all know that fossil fuel is going to deplete and then it will exhaust in few years. So we need to search for the alternative fuel. biogas is possessing a major opportunity of meeting the energy demand. Second is it reduces the energy requirement for the wastewater treatment plants, because for operating a wastewater treatment plant it requires a lot of energy. So if biogas is produced in addition, it can meet this energy demand. Then third is it replaces the chemical fertilizer. Then fourth is it replaces the LPG cylinders that we are using it from the fossil fuels. Then finally, as I indicated, it is a zero waste technology. So whatever you are putting in, everything will come out as a product. So we can call it as a zero waste initiative. I would like to summarize the things what we have discussed in today's lecture. The first thing is the list of waste materials what we can utilize for the biogas production. Second is the different operating parameters like pH, temperature, carbon nitrogen ratio, and then solid waste ratio that we need to maintain for the better anaerobic digestion. Then third one is the anaerobic digestion, which is the important topic in today's lecture. It comprises of the 4 steps like hydrolysis, acetogenesis and then methanogenesis. Then next is the reactor, that is called anaerobic digester, in which the anaerobic digestion will take place. We have also seen some different types of anaerobic digesters, and finally we have seen some of the advantage, also the applications of this biogas. We are coming to the end of this today's lecture. If you have any doubts, you can put your doubts in the Wikipage forum. Thanks for listening. Meet you in next lecture. Thank you. Have a nice day.